Hi guys, hey, 9:30, we're right on the dot. So, this is just a catch-up stream, for those of you that missed yesterday, because we had a very, very big delay last night. So we're just going to do a really quick one because I think the exam's in the morning, so we'll just, wrap it up hopefully before 10:45 so you can get some decent sleep. And so like yesterday, if you just want to chuck in some questions in the chat, then I'll attend to it. All right, so try not to don't ask me to go over a particular exam question that I recently did a video for. You can just look it up on the YouTube channel. All right, so so chuck a question in the thing, please. So this is for the year 11s. I understand the level 3 chemists have an exam tomorrow as well, but I don't know, I have a feeling that the year 11s probably need a little bit more help. General tips for mechanics: learn to use the formula. You get given the formula sheet. Look for the numbers, look for the unit, and substitute numbers into the equation. Make sure you know which one you're using, and then don't type the wrong answers in your calculator, and don't copy the wrong answers from the calculator. If you can do that, it's an easy 4 credits. Okay. What's the inheritable and non-inheritable variation? So we kind of did that yesterday. Inheritable are things that can be passed on from one generation to the next, whereas non-inheritable, say for example, if you hit the gym quite a bit, do a bit of weights, does that mean if you were to have children, they're going to be as big as you are? Not really, because that's not inheritable. Okay, so those are some just examples. Can you explain sexual reproduction? So again, did this yesterday. Sexual reproduction results in variation because of two things. One is meiosis, and the other bit is fertilization. Okay, so just briefly, because you can look up at the NCEA websites to look for some odd answers, but just to help you with understanding, just remember, meiosis, you create unique gametes, so all of the 6 cells that are produced are going to be unique. Okay, and then these gametes are all different, because of crossing over independent assortment. They're all going to be unique. They have half the number of chromosomes, and then each parent's gametes combine together, and which one of those gets to fertilize is random, and that results in a complete random offspring. Okay, so in terms of the genotypes. Okay, so that obviously increases variation. And what is meiosis and when does that occur? During or before after cell division. Sarah, meiosis is a cell division, so it's not before or after, it is a cell division. There are 2 types of cell divisions. One is mitosis, one is meiosis. Meiosis is producing 6 cells. Okay, so it happens in the reproductive organs. Meiosis occurs, you start with one cell. Let's say we did this yesterday. You start with one cell that has got, let's say, 4 chromosomes. Then after dividing twice, you are going to produce 4 daughter cells that each have half the number of chromosomes. Okay, so this one has 4, this has 2 chromosomes because they all have half the number, and that is a cell division process. Okay. A quick way to write ionic compound swap and drop. Priya, if you ever write swap and drop in the exam, you're not going to get any mark for it, because as a marker, I can tell you that right straight away, the moment we see swap and drop, cross. You don't understand what you're doing. Okay, you can do swap and drop when you're trying to figure out the thing, but if you write swap and drop as an explanation, you're not going to get anything for it. Okay, so just a heads up. What is the difference between variation and mutation? Genetics will help. Okay, so with Ivy, what's the difference between variation and mutation? Variation, like say for example, if you think about all the human beings except identical twins, like say if you think about you and your parents or you and your siblings, we are really identical to within the family, but then we're slightly different. Why? Because of variation, because of the slight difference in the genetic makeup. And so that's variation. Okay, and mutation is a cause of variation, because what causes variation? variation, which is a difference in the genetic material, which results in physical, differences in physical structure. So variation is a level 1. This is from sexual reproduction. The other one is mutation. Okay, so mutation is just due to radiation and whatnot, and then there's a sudden change in your DNA base sequence. You make a different protein. You have a different allele. Then you suddenly have a unique physical characteristic. Okay. Are there more H3O plus atoms? And that's wrong. H3O plus ions. Anything with a plus charge is ions, or negative ions is an ion. And there's more because. Okay, so are there more? Okay, so just remember in acid. Again, this is yesterday. Acid, neutral, basic. So acid in just do this I think I did H3O plus because you're only year 11s, don't use that. Just use H plus. In a neutral solution, your H plus equals OH minus. In acidic condition reaction, your H plus is greater than the hydroxide. Oops. And then the bases, the H plus is greater than the H plus. H3O plus is more correct, but you're on year 11s, you don't need to know that. Is inheritable through sexual reproduction? Is inheritable? Yes, if it's inheritable, obviously it's done through reproduction. Otherwise, how would it be passed on? Because we're not clones from our family. Do I have to include the full process of meiosis and mitosis in your answer? Mitosis you barely need to mention because, like I mentioned multiple times, this topic is called. Well, one of the topics is called genetic variation. Mitosis doesn't result in variation, so it's something you don't really need to look at. Meiosis, you don't need to know how to draw the diagram. You just need to know meiosis. One cell produces 4 daughter cells with half the number of chromosomes. They're all unique because of crossing over. They swap bits and pieces of the information, and then they line up randomly, and then so that creates a new unique gametes. Can I run through some equations? Jack, can you be a little bit more specific? Give me a topic, and then I can give you some equations that we can, concept that we can do. Kira, when you're doing calculation for physics, and it includes calculating the height, do you calculate the vertical height or the horizontal? Well, if it makes sense. Well, if you're talking about lifting things up, like say, if I have this box, yeah, and I want to pick up this box by, like, I don't know, 2 meters, how much work am I doing? And then you go work equals force times distance. And you may go, oh, what is this distance? This distance is how much it's traveling. In this case, it's traveling upwards. So that distance will be 2 meters. And what is this weight? This force? This force is the weight force, because that's how much work you need to do. Okay. And when they specifically talk about height, it's vertical, like Sarah has said. Distance, yep, in your 2020 around you show the formula F equals mgh, so all of a sudden you said the height is 20. It's probably because the answer is in the question. So this is answering the 2020 mechanics video, because it would have been somewhere in the question if you read the question. Okay. What's an easy way to get the symbol for salt? Adam, what is a simple way to get? So I'm assuming like say for example, if I have hydrochloric acid plus hydrochloric acid plus zinc hydroxide, for example. Right, so I'm assuming that you can do this. You know that hydrochloric acid and zinc, the zinc, and then the hydrochloric becomes chloride, so this is your salt, zinc chloride plus water. All right, I'm assuming you can do this. All right, now if we were to do the equation is just a translation of what we just wrote. So hydrochloric is HCl, zinc hydroxide is ZnOH2, and you may go, wait, how did you figure this out? Am I supposed to memorize that? No, you're not supposed to memorize it. You're supposed to figure that out using the table of ions. Zn is 2 plus, OH is 1 minus. That's on your table of ions, and you look at this, that's 2 plus, that's 1 minus, they don't perfectly cancel. I need 2 OH minuses to get rid of 1 Zn 2 plus, so it's ZnOH2. Where does that 2 come from? Because you got 2 OH minuses. What do I make here? Zinc chloride. What's zinc? Zinc is Zn 2 plus, what's chloride? Cl minus. There's 2 plus charge, there's 1 minus charge, they don't cancel each other out perfectly, so I need another Cl minus, so that means I have 2 Cls, so ZnCl2. Alright, so this is ZnCl2, water is just H2O. Now that's merit. If you want your excellence, this is 2 Cls, this is 1 Cl, put a big 2 in front of that, and then once you've done this, you have 2 H's, you have 4 H's, and there's only 2 here, so put a big 2 in front of that. Okay, so the word equation is achieved, the symbol equation is merit, the balancing symbol equation is excellence. Okay, so there you go. Then Jack, there's an excellence question for you, mate. And then another excellence question that a typical excellence question, Jack, for excellence for acids and bases is like, say for example, why does sodium chloride have why is this 1-to-1 ratio and sodium oxide is 2-to-1 ratio? Why is that? Explain using the table using the charge and the electron transfer. That's one. And then another one could be why does sodium becomes +1 charge ions and then fluoride has a -1 charge? Why does that happen? And then you can have excellence question of rate of reaction. Rate of reaction, you know, why does increasing temperature result in increased rate of reaction? Why does high surface area increase rate of reaction? And another type could be a pH question. Like say I start with hydrochloric acid, you start off with red, you know, in universal indicator. I start adding a base inside, go through all the color changes until normal color changes stop, until normal color changes occurs, go through all the pHs, go through all the colors, go through all the ion concentration comparison, and that's your excellence. Easy peasy. All right, now what's the next one? Level 3 chem, good luck, eh? Level 1 is a joke compared to level 3. Q equals MC delta T. What is continuous? That's continuous variation is not something you really get asked in level 1 genetics anyway. Like, it just means it's more of a, now if I remember by definition correctly, it's to do with the like it's on a continuous spectrum, if I remember correctly. Like some, like say for example your height, you know, because it's a range of height. Whereas hair color, there's only a few, you know, a certain number of hair colors, you know, you don't really need to worry too much about that. How do you explain ratio of ion like CaCl2 and explaining the electrons? It's easy, CaCl2, all right? So this is made of Ca2 plus and Cl1 minus, yeah? So the first part you can do, you can just say, oh, this is 2 plus, this is 1 minus, I need 2 Cl minus per 1 Ca2 plus to balance the charge. To form a neutral ionic compound, so you need 2 Cl minuses per 1 Ca2 plus, and the other bit that you can say is that Ca, well not Ca, this calcium atom is 2882, chlorine is 287, so this needs to lose 2 electrons, and this can gain 1 electron to become stable, to have a full outer shell, to become stable. So this needs to lose 2, but this can only gain 1. So you need 2 Cls to gain 2 electrons from the calcium in order to form a neutral ionic compound. All right, very, very straightforward. Could you do hard balancing equation? There's no hard balancing equation. This is level 1 science. Trust me, there's no hard balancing equation. Most of the time, you just multiply something by 2, then you're done. Ivy, can you explain the pedigree chart? Pedigree chart, I did one yesterday. It's more just about, so let me find one for you, shall we? Pedigree chart, because you will not be the only one, because it's an excellence question, so might as well go through that. Just bear me for 1 sec. Let me find a past exam paper. Genetics, let's do 2014. All right, here it is. So this is 2014 exam. Is it a pedigree chart? Blah blah blah. Don't care. Oh, no Punnett squares. No. Oh, here it is. All right, let's just crop this. Okay, where's my snip? I'm just gonna snip this bad boy. Alright, and let's go back here. Alright, so here it is. So this is an exam question, and pedigree charts. So like I always do, I don't even show you the question, because if you can't do what I do, you can do any questions. So in the family tree, people can roll their tongue. It's displayed as us. It's pretty cute. People that can't, like me, you do like that. Okay, and then use this letter, use capital T and lowercase t for tongue rolling. So anyone that can roll their tongue has a dominant trait. Anyone that can't roll their tongue is a lowercase t. Okay, so that means these guys are all going to be small t, because they can't roll their tongue. That means they must have both of the recessive alleles. So go to the diagram. Where are they? So this is small t, 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 small t. Like without even reading what the question asking you to do. Like you already figured out most of the question anyway. This part, all right. So if they have rolled their tongue, that means they have at least one capital T. The other one we don't know. So number one is going to be, let's get rid of this. This is big T something, big T something. This is big T something, big T something, big T something, big T something, big T something. All right, and most of the question is just asking you to figure out, oh, what is that something, So for number 3, let's use this example. If you look at number 3, like if we're trying to figure out what is the genotype for number 3, this can be easily done, because if you look at the offspring of 3 and 4, number 8 is small t, small t. So how the heck are you going to get a child with small t if you don't have small t's in both of the parents? So that means number 3 is going to be big T, small t. Number 4 is going to be big T, small t. All right, because you can only, the 2 small t's means that you have to get 1 small t from each parent, but we know for sure that each of them must have a capital T, because they can roll their tongue. So that means the other allele must be small t, and then you can just do a Punnett square like that, and just to show what it is. Okay, so this is your logic of doing Punnett squares, and that's going to make your life a lot easier. Just don't even worry too much about the question. Once you get a question like that, just make sure you identify which one's a dominant trait, which one's a recessive trait. You can start with a pencil and start doing those. What is the easiest way to remember collusion theory? Just remember particles must collide with sufficient amount of energy to have a chemical reaction. That's pretty much what we need to remember. So they need to collide, they need to hit each other, they need to hit each other with sufficient amount of energy to break the existing bonds to cause a reaction to take place. When do you put elements in the brackets? And so, like say for example, if I do zinc hydroxide again, why do I do it like this? Why can't I just do it like that? Why would you get marked wrong? Well, yes, of course you get marked wrong. Then this is the reason. Now we knew that for zinc hydroxide, this is 2 plus, this is 1 minus. So can you see there's 2 lots of OHs? So if you do it like this, if you do Zn OH2, if you do it like that, can you see there's only 1 O? You suddenly lost an oxygen. Because if you count here, there's 1 OH, there's 1 OH, there are 2 O's, there are 2 O's and 2 H's. Okay, so what we want to do is that we want to multiply, we want to indicate we have 2 lots of OHs, and the only way you can successfully demonstrate that is by putting the bracket. Okay, now would you get marked wrong if you did it like this? Like say for example, I've seen excuse me, I've seen people doing this, like say zinc chloride. I've seen people doing this in the exam, and we've been told as markers that that is okay. All right, so we accept that because it's like in mathematics. Like, would you get punished for writing 1x? Not really, but it's very bad convention. But we just don't mark it wrong. Okay. How do you define a recessive allele? Recessive allele is just the allele that doesn't get displayed. Recessive allele is only displayed if it's the only allele available. Okay. The only bracket. Wait, that's a complex sign question. Wait, where was I? The only bracket complex signs, so that's a level 3 chemistry question. Yes, you have to bracket complex signs. Particles must collide. Yep, done that. What's the smartest way to write the papers like the water? That comes down to personal preference. One tip for you guys doing exams: start with the ones that you're most confident, and if you come across a question that you're gonna struggle with, just leave it. All right, because what tends to happen with especially teenagers is that you can like being able to do an exam well is actually a really good technique. It's a really important technique. Don't get hard stuck on the question and and beat yourself up to it. Like you're like, oh, I know the answer, I just can't remember. Just if you give yourself a couple minutes, if you don't know how to figure it out, just leave it, all right? And you come back to it. Just make a note on your script, fold the paper so you're gonna go back. But no point dwelling on the question that it's gonna suck away a lot of your time and it's gonna tilt you. Just move on. There's plenty of other opportunities. Like I mentioned before, if there are 2 excellent opportunities in one question, you only need one for E7. So there's no point, spending most of your time and then just, emotionally and psychologically, hating yourself because you can't solve it. Just move on. All right. For order, again, that's to your personal preference. Resistance, yeah, there you go. Can you do Mechanics 2019C? It's on my YouTube channel. Look it up, mate. It's there. It's there. What's the difference between sulfide and sulfate? Adam, that is a fantastic question. You have no idea, my friend, how many people get confused with sulfide and sulfate. Oh, geez. So many people get this wrong every year. Sulfide is S2 minus, sulfate is SO4 minus. So if you have a salt, like say sulfuric acid with zinc, you make zinc sulfate. You don't make zinc sulfide. Okay, sulfide is not a very common salt. So that's an excellent question. All right. Integrals. Why are we talking about mathematics here? How do you remember ways to write word equations? Acid plus base give you salt and water. Acid plus base give you, there are some, like I've seen people do this. Like, is this what people do? Cows or what's the? I don't even know how you do it. I just remember acid plus base. I mean, I'm old school. I didn't learn this with all the fancy acronyms. I just go acid plus base give you salt and water. Acid plus whatever carbonate gives you salt and water and carbon dioxide, because, and one way to remember it, because this is carbonate, has to go somewhere, so it turns to carbon dioxide. That's what made fizzy drinks fizzing. And the other one is acid plus metal, gives you salt and hydrogen gas. So just memorize them. I don't, that's pretty much what I have to offer. Some of you answering questions. Unit for order formula. I did that yesterday. Let's find it again, because people ask for it. NCEA 1 science subject resource. Give me a sec. I'll find that thing again. Mechanics. Let me just get my thingamajiggy. Yep, copy that, goes here. Alright, so this is the equation sheet that you get in the exam. I'm going to make it a little bit larger this time. So this is the mechanics resource sheet. You get this at the very top, so make sure you know which one's which. This is speed. Speed is meters per second. This is distance, meters divided by second. Acceleration is meters per second. Speed is meters per second, like the one on the left. Time is seconds. Force is measured in newtons. Mass is measured in kg. Acceleration is m is -2. Pressure is Pascal. Force is newtons. Area is meters squared. EP is joules. Mass, m is kg. And g is gravity, which is 10. Change of height, this is in meters. EK is kinetic energy, so joules. This is kg. The mass, V is velocity, so meters per second. Work is joules. This is force. Distance is meters. G is given to you. Pressure, this is not pressure, this is power. Power is in watts. Work is in joules. Time is in seconds. So there you go. Next lot. Key excellence question from each paper. Acids and bases, formation of ions, the ratio of the ions in the compounds, rates of reaction, acids and bases, balancing equations, the pH colors, ion concentration, use of observation to determine what you got. So those are your acids and bases papers. Genetics, pedigree charts, guaranteed. Why is variation important? Guaranteed. And then the rest can be fluctuating a little bit. Sometimes could be Punnett squares, like could be mutation, could be sexual reproduction. Could vary, because I can make them achieve questions. If I was a examiner, I could make them any of those questions excellence. Mechanics, pressure work calculations normally can go beyond to excellence. Those speed time graph linked with net forces, those can go up to excellence. Pressure calculation and explanation can go up to excellence. EP equals EK, conservation of energy, can go up to excellence very easily. Okay. And how is sodium hydrogen carbonate a base if it has hydrogen? Well, yes, it has hydrogen, but the thing that we're most interested in is the carbonate. Okay. Yeah. So carbonate, that makes it basic. For fertilization, do we just talk about how it is random? Which, yes. So Nozi, Nav, I think that's how. Hopefully I didn't butcher your name. So just remember, each of the gametes are unique. And then which of these unique gametes get to fertilize again is random. So that's why you can have many siblings in the female, but they're all going to be slightly different, because every time the fertilization occurs, the sex cells is different. Okay. Looks like a lot of you answering your own questions. That's really good. Powers of swords, I've done that. When we start the mechanics topic, symbol, I'm not going to go through any particular topic, because I've done if you're more into, because this is really last minute, if you're really into like a more structured tutorial, you can look at my previous exam questions, or look at last year's Q&A before the exam, which was quite good. One day the zygote has different allele combos. Yeah, they have just a unique combination. In excellence questions that don't have bullet points or bolded, do we have to write about things that they didn't ask us for? Jazz, that's not, they would have told you, you're not expected to write things that are not being asked. So first of all, read the overall question, because a lot of things, like, this is actually a good point, because like blah 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 blah, this is a question, and they will go, in your answer you should blah 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 blah. There's bullet point 1, bullet point 2, bullet point 3. All right. Now normally they put their big question there, so they'll have a big question there. So make sure you answer that, because a lot of people get fixated on this, and then they've actually forgot what the question's asking them to do. Okay, so make sure you cover all of them. Do we have to talk about the period in the group? No, you don't need to. If you do, it's just a bonus. If you didn't, it's not compulsory. And speed, velocity with direction. Velocity is speed with direction. You got the other way around. Can I explain EP equals EK? So have I almost caught up with chat? Oh, almost. EP equals EK. So etymony, I think that's how I pronounce your names. Like, say for example, if you hold up a pen right now, yeah, and then if you hold a pen to a particular height, and if you let your pen go, it's going to fall to the ground. Yeah, so if we assume EP equals EK, like say your pen is a certain height from the ground, you can calculate how much gravitational potential energy it has, because you know how much your pen weighs, you know how high it is, you can calculate EP. Okay, so if we assume all of that energy is going to be converted to speed, like the movement, so that means all of the EP will be converted to EK, and then you can calculate the theoretical speed. But then in reality, your pen will never ever be able to travel at that speed, because if you let go of your pen, it's going to drop, you're going to hear it bounce on the floor, it's going to make a sound, you're going to lose that energy, some of the EP to heat, some of it to kinetic, some of it to sound, which means not all of the energy is going to EK, so you're not going to get to that speed. Okay. What are common mistakes in mechanics? Misuse of numbers, misuse of units. In pressure, people forget that people have 2 feet, elephants have 4 legs, cars have 4 tires. You get the area wrong. Reading the graph, speed time graph, guys. When you have speed time graph versus distance time graph, when you have a graph that looks like this, they mean completely different things. Oh, this is a horrible graph, right? For example, like that. Speed time graph, this is a straight line, so this is a straight line, this is a straight line, this is constant speed. This is constant speed, whereas this is stationary, right? You know, things like that. And the only way you can tell which is which is by look at the unit. There you go. What a common mistake. Yeah, did that. All right. What are some excellent questions for genetics? So Stacey, I kind of mentioned that already. So pedigree charts, guaranteed. Need to draw Punnett squares. If you go into, I kid you not, if you go into the older exam papers, go to question 3. It's always some fancy context like trees, butterflies, mushrooms, whatever. Doesn't matter what it is, and they always go. So why is it important for these species to have variation? And you just say, oh, because it's important for them to have for the population to have a good variation of the genes, because if just in case the environment changes, a disease happens, some of the population will be able to survive, pass on the genes, and then the population will remain successful. Blah blah blah. Okay. How to draw forces and label them? How do we draw them? Lunar. So this is, if I have a car, that's awesome car that is. If I have a car going forward, this is a thrust, this is friction, this is support, this is gravity or weight. So the way I've shown you, can you see the thrust force is greater than friction? Because my graph is indicating that. So that means the net force is going to be bigger than 0. It's going to go to the right side. That means it's going to accelerate to the right. Okay, simple diagrams. What happens in crossing over independent assortment? So Sarah, for crossing over, just remember they swap bits and pieces of information. Independent assortment, they just line up randomly. Done. Just put that in. Is gravity a force of or acceleration? Gravity is acceleration due to weight force is acceleration due to gravity, and gravity is the pull. It's a force from the center of the planet due to the large mass and the orbiting, pull, sucking things into the middle, into the core of the of the core of the planet. Yeah, so it's more of a force if you think about it. Yeah, but then there's acceleration due to gravity. All right, so gravity is a force. And if you were doing chem and physics, just pick one, mate. Go with the one that flick a coin and go with that. What do forces do? What do forces look like on things in air? Well, in air, that's actually a good point. So what I've done is like on the ground. If it's in the air, like say an airplane, this is still thrust. You can say engine force if you have like an engine. You can still go with friction. It's not wrong. You can say drag if it's like in the air. This is going to be again gravity or / weight. And while in the air you have no, you see, this is where it gets a little bit tricky. So you got to think about the context. So what are we talking about? Like a helicopter. So helicopter have a bit of thrust going upwards. So you can talk about lift. So this is talking about things that are in the air. But then if things are sitting on the ground, you have just support force. Spencer, what is collusion theory? Collusion theory is when it just means that in order for particles to in order for a chemical reaction to collide, particles must hit each other at the correct orientation with sufficient amount of energy. Write that in. That is one easy achieved tick right there if they ever ask you that. Okay, one achieved. You only need 8. So there you go, 12.5%. What happens if you round my answer to? In level 1, we don't punish you too much for rounding. You should be safe. But if they said round, then you do need to round. But otherwise, it's fine. Can you do some example for balanced symbol equations? Symbol equations. Let me just find balancing symbol equations. magnesium hydroxide plus nitric acid makes MgNO32 plus H2O. I'd say you can give it a go balancing it, maybe pause if you want to give it a go yourself, but this is what I tend to do. Can you see there's 2 NO3s? There's only 1 NO3, so I need to put the 2 there. There's 1 Mg, there's 1 Mg, so that's fine. That's fine. So there's two H's in there, 4 H's on the left. There's only 2 H's. Put the 2 there, and that's it. All right. That's as hard as it gets for level 1 science. Sarah, buoyancy, yes, in water buoyancy is definitely good. Yeah, but I don't think I've ever seen the question underwater. Yeah. Could you define air resistance? Air resistance is just it's like friction, but in the air. It's just you're trying to, even though you can't see, but when you're trying to, there's air particles all around us, and when you're trying to push against them, they will slow you down. So it's like air resistance. Okay. How could a change in gene result in phenotype? Well, what is the gene? Gene is the DNA blueprint that tells the body to make certain types of proteins. So if the blueprint has changed, you're going to make a different protein. So if you make a different protein, you're going to look different. There you go. All right. How are we going? Everyone's having a good go at yeah, mass is good. Yeah, that's good. Yeah, 13 here. Just want to say thank you. Oh, no problem, bud. Just good luck tomorrow, you know. Yeah, I hope the year 13s get as fair paper as the year 12s. The year 12 paper is very, very fair. So I hope you guys are as lucky as the year 12s. What does 10 newtons per kilogram mean? So Daniel, 10 newtons per kilogram, it just means for every 1 kilograms of mass, that's equivalent to 10 newtons of weight. Okay. If you're not into that much physics, just remember gravity equals 10. I don't even care about the unit. You know, I'm being very pragmatic here. Okay. Oh, oops. I've done all the questions. Are there any more? Are there any more? What is a disease inheritable? Well, it depends on what sort of disease we're looking at. If you're looking at a genetic disease, you know, one I've had, you know, when we had my son, you know, they went through like during pregnancy, the mother would go through like Down syndrome tests and all those sorts of tests because they're genetic diseases that can be passed down. So you do need to, some diseases are genetically inheritable, which, you know, those parents will know. Yeah. Wait, did someone donate? Oh yeah, someone donated. Oh, thank you very much. $5. There's coffee there. Thank you, Samuel. Do I have a drawing tablet? Yes, I do. And as you can see, my handwriting is still shite. That's just, I can't write very well. That is what it is. Could I go relationship? Oh, one more time, James, just for you. All right, so ready? DNA. I'm gonna say it. I'm not gonna write it. Just remember. Ready? DNA, genes, alleles. One more time. And I'll chuck in some genotypes and phenotypes for you as well. All right, very you know, chuck in those for free. So the DNA is a double helix structure that, you know, contains all the genetic information. A gene is a section of the DNA which codes for one physical characteristic, whereas an allele is the alternative version of the same gene. But then, you know, so they're different versions of the same characteristics. And what makes alleles different from each other is that they have different base sequence, which is ATCG stuff, right? So the order of the base sequence is different. So ATCGs, the base sequence is different, which means they make different proteins, okay? So when every, you know, unique human being, we have unique genotypes. Okay, so the genotypes is a combinations of the alleles that you get from your parents, one from each parent. And then the phenotype is what your genes would physically be represented from the formation of proteins. Okay, so that's that one. Okay. Pablo, no problem, but hope your exam went well. Would there be questions on cloning? You never know. I haven't seen one recently, to be honest. I don't know. Cloning it's quite controversial. Ethically very, very wrong. So we'll see. Like I said, the bio paper, they will chuck in a new context every single year, but the question is going to be the same. Purpose of DNA just contains the genetic information. No to me. So is a mutation a change in the base sequence? Yes, you're right. It's a change in the base sequence, so that completely changes the gene, which means the change of allele changes the genotype, changes the phenotype as a result. What are the benefits? What are all the benefits? All right, plasma. You look asking about. So the benefits of asexual reproduction. So as you know what you're gonna get, because they're just clones. So if you see the parents, the siblings. So the offsprings are gonna be similar, well, exactly identical. So you know what you're getting out of. So asexual, the outcome. It's gonna be less time consuming. So because they just asexually reproduce, they don't need to look for partners and they don't need to invest a lot of energy. But the downside is there's no variation. So if one catches an infectious disease, and they all die. All right. Whereas sexual reproduction, the downside is you have to find someone to reproduce with, and that involves energy, blah blah blah. A lot of investment to go into that. And that's why if you look at all the species in nature, like all the birds in particular, the male birds are the ones that are really colorful. Like say something, the example that comes to mind, like peacocks. Peacocks are very, very colorful, but it's only the male that are colorful because they want to attract the females, and so they need to invest a lot of energy into that. But then they put themselves at risk because every living thing can see you from a mile away because you're so colorful. Okay, but so what's the upside? Genetic variation, which results in success in the species. All right. Yeah. There we go. Very helpful. No problem. Anything up? Precious amount of force exerted in an area. Yep. Your science serious, right? What is change in base sequence and change in base sequence of a gene is exactly the same thing. So it's the same. You're saying the same thing. How to answer a question when they're asking you which takes more power on the ramp, or just taking step by step. So remember, if I say for example, if this is a bike ramp, so if you get the bike up here, or you just lift the bike up here. So at the end of the day, the work that you're doing is the same, because what is work? Work is how much energy have I done to get this bloody thing up there. So if I get the bike to this position, to this particular position, that means doesn't matter if I walked up the ramp or lifted it up, the bike at the end would have the same amount of EP. Yeah, let's say for example, if again back to the pen example, if you put your pen on the table, and you hold it up, and you can pick it up really quickly, or you can pick it up and then swing to the left, swing to the right, swing to the left, swing to the right, then do a rotation, do a 360 twist, whatever, it doesn't matter. But then you get to the pen to the same height. So it doesn't matter which method you do it, the pens at the end of the day are going to be at the same height. If they are at the same height, they have the same EP. Okay, so when you do P, power equals work divided by time, work is going to be the same. So what is going to be the factor here is going to be the time. If you do it quickly, then you have to invest more time. If you do it in less time, it's more powerful. More time, less powerful, for example. All right, okay, so that's right there. But I think if you're talking about lifting, putting this up there, or go up over here, and then you can talk about work equals force times distance. And then if you're putting up a ramp, you're actually spending less force. You're spending less force pushing it up because it feels easier, but because you're traveling more distance. Okay, you're traveling more distance, so that makes up for it. Or if you're picking up the bike, it may feel like a lot of work at the for that brief moment, because you're investing in a lot of work. Investing a lot of force, but then you are only carrying up a very short distance. Like think about a bike ramp. A bike ramp could be like 10 meters long, but if you go to the end of the ramp, pick up your bike, chuck it up there, you're only lifting it up by what, a couple of meters. So it's that's how you can. But at the end of the day, the bike has the same same amount of work. Yeah. There you go. So what's happening here? Why are we talking about? How would you describe variation? Variation, just differences in the same population. Boom, done. What school do I teach at? I teach at Botany, BDSC. Kingdom. I think it's either Koru or Britain. One of those. Good luck tomorrow. Wait, why are you doing exams, Kingdom? Aren't you triple E for UEG? But doesn't matter. How does variation actually make you immune to a disease or virus? Plasma variation doesn't make you immune to a disease or virus. Variation increases the chance that someone in the population is immune to the disease or virus. Okay, so it just increases the possibility. It doesn't mean you, like me in particular, is going to be immune. It doesn't mean you in particular are going to be immune. It just means out of the how many people now? 7 billion people on the planet, is bound to be someone that's going to be immune to the disease, and then they can survive. Yeah. Lewis, if you have any level 3 chem question, yeah, maybe towards the end, once all the year 13s, once all the year 11s have buckled off, I can help you with some level 3 stuff. Sure. How does sexual reproduction help increase the survival rate of a species with those COVID-19 viruses? Yeah, well, like I said, if someone's bound to survive and pass on the genes, and then make sure the species continue. What do you need to know about meiosis? Just need to know, Sahib, what do you need to know about meiosis? You make unique gametes, which is half number of chromosomes, and then they're all unique because of crossing over in independent assortment. That's pretty much it. 4 you need to, yeah, done there. Any last minute tips? Just know your definitions. All right, that's the thing, not just with level 1 science, level 2 bio, level 3 bio, university bio, learn your definitions. Okay, you have to, when they say link these words together, if you don't even know what they mean, you're not going to be able to do well. Okay. What process, meiosis, fertilization, cause variation? So Franny there is pretty much got you sorted. So how does it cause variation? Because the gametes are all random. How would you word and structure in the exam? So I'll just say, well, meiosis is produces sex cells. So during the cell division process, one cell divides into 4 daughter cells with half the number of chromosomes, and then they're all unique because of independent assortment, where they're randomly aligned and sorted, and then the crossing over happens, which means they swap bits and pieces of information. So the gametes are all unique, and then the fertilization occurs. So one of these gametes are going to be randomly fertilizing another one from the opposite gender. Therefore, all of the offsprings are unique. There you go. Do we know the definition of population? No. You don't need to know that. Yeah, it's fine. Like a lot of the questions I've repeated. It's okay. It's fine. That's why I'm here. Is force and distance inversely proportional? Well, actually, it depends on what formula you are looking at. Yeah. If you need to go to sleep, just go. All right. Just go. Haplo is half. Yes. Have you want to? I'm not going to go through any paper in particular because, like I said, there's plenty of videos for year 11s on my YouTube channel. Just go through those. All right. I'm more than happy just to answer individual questions here. What's Alicia doing here? Isn't she year 10? Are you making this year's papers? Am I making this year's papers? I can definitely tell you I'm not making the year's papers. I'm marking the year's papers. I'm not making them. Right, so that's 10:20. Are there any questions? Stress about if there's have already gotten numerous E's in science and past level 1. I mean, it's really up to you. It's what you strive. Like, do you want to get a lot of excellence credits? Do you want straight E's? Do you, it's really about you. Like, what motivates you? This is what motivates you. If I make the papers, can I give you the answers? Definitely not to you, Kingan. I'll give it to everyone else, but not you. Sorry, but if I were to rephrase the question, how does the change variation actually make you immune to a disease? How does variation in gametes? Okay, so plasma, what it means is that, again, because we're all different, our genes are different. Like say, could be like say for example, a new virus that's more deadly than COVID. Like say, I don't know, something that comes up and attacks a particular part of the DNA. For example, I don't know, but then some people may not have that particular trait, and then they are immune. So it just by chance, it's all random. How many credits do you need for course endorsement? From memory, on the non-COVID year is 14. And you have to get them from both internals and externals. They can't be all from externals or internals. I think it's 14. Yeah, 14. Sounds about right. Want to go to the Air Force? Well done. Yeah, Air Force. Good on you. Good career. Crossing over crosses chromosomes. So crossing over is kind of like this. When these chromosomes line up in the middle, and when they do this, they swap bits and pieces of the information. Yeah. If you can't answer the actual answer, that's okay, but you just can't do something. If you can't answer the actual answer, is it okay? We just define the term. So if you, like say for example, if they've given you something, you can't, if you leave it blank, you're never going to get it right, all right? Like say for example, collusion theory, one people always ask, if you get one of those rates of reaction questions and you just have one of those moments, you just can't remember anything, put down the collusion theory in there. You can just say collusion theory is when the particles, they must collide with sufficient, at the current orientation with the correct, with sufficient amount of energy. Chuck that in. You'll be surprised how often we give you an achieve for that. Okay. What is a simple definition for zygote? Zygote is just when two gametes of the opposite genders combine together. So zygote. Yeah, there's my mod, Tony. Year 11's have it good. Yeah, I know, he's crying. Year 12 physics. Does exam matter that much if you're year 10? Huh? Well, if you're only year 10, I suppose. I don't know. It's really up to you. Like, you could be year 11 and it doesn't matter to you. You don't try hard. It's yeah. The screen is not frozen. I'm not writing anything. I'm just saying it. Do you need 3 excellence in externals and internals to get excellence endorses? Fine, you can merit. So to get endorsements, you need, if you get 3 excellence in externals, and then if you get 1 more internal excellence, then that should be guaranteed endorsed. Yeah. Are the 4 daughter cells identical? No, they are not identical. That's the whole point of sexual reproduction. They make unique daughter cells. They're all different. See, the good thing is that I really like what a lot of you are doing. Like, if you see someone asking a question, and then if you can answer it, and if you can explain that to that person, that means you understand it. And the best way to know that you know something is that you teach someone. So it's good to help each other out. So that's a really good thing a lot of you are doing. Yeah. What mark does balancing symbol equations give excellence? So Abdullah, if you just write the words, achieved. If you can write the symbols, all symbols correct, but merit. If you can balance it, excellence. Yeah, you get a certificate. Yeah. If you go to the one paper, if I didn't want to do genetic, even hand it in. If you don't do the paper, just leave it completely blank, it will be noted as a SNA, standard not attempted. Yeah, just don't write anything. Okay, just don't write anything on it. Less than 12 hours, yes indeed. Any more questions before I think Lewis in the year 13 is probably still waiting. Lewis, are you still in the chat? If you maybe want to chuck your question in there, I'll just see what it is and maybe I'll give you a hand if the year 11s are happy. When you go in for examination with all 3 papers, yes, they'll all be set on your desk. So how do I know it's reaction spontaneous or non-spontaneous? So this is year 13, okay? So don't panic. All right, so spontaneous or non-spontaneous. So you want a negative change of enthalpy, you want a positive change of entropy. All right, so negative change of enthalpy, that ensures the environment. This is year 13. Don't panic. Oops, what just happened? Oops, my pen just spazzed out. Let me just get rid of that. So negative change in enthalpy, that means the surroundings entropy increases. And then if you have positive change in entropy, that means the system's entropy increases. That means the whole system's entropy increases. Yeah. Will I be at school tomorrow? I'll be at school. Yes. What time? Probably 8:30ish. Okay. Hey, Hunter Harrison. This will be you next year. Are we supposed to know names of catalysts? No. You don't need to know the names of catalysts. Everyone gets exams in plastic pockets. You need to reproduce. Yes. Have you guys not set any exams yet? I mean, you're year 11s, but surely you've done some maths and English and classics or history or whatnots. It sounds like a lot of you have not done any exams. You just go in. You don't write your name or anything. Your code. Your name is your number is on top of the paper. Yeah. What do I do with your phone? Leave in your pocket. Don't worry. You know, if you've done the study and you know it, you will perform. Okay, you will perform. A really good way, like if you grinded past 3-4 years of exams, you should be okay. That's just, you know, it'll be okay. Just don't panic. You'll be fine. Carbonate was reacting with sulfuric acid. You said this will be fizzing, but no reaction for something oxide. There will be no reaction. Yeah, because plasma, carbonate and acid produce carbon dioxide. Acid plus base gives you water and salt. There's no carbon dioxide. There's no fizzing. Yeah. Could I briefly explain meiosis and fertilization? So again, Daniel, meiosis is this cell division process that occurs in the sexual reproductive organs, where you make sex cells. All right, you make sperms, you make eggs, you know, and then what happens during meiosis? You make all of the sex cells are unique, okay, and they have half the number of chromosomes. Why is it half? Because you need one from each parent to combine together to give you a normal child. And fertilization is just, you know, the process of whichever one of these gametes gets, you know, chosen to be fused into a zygote, into a baby, but that's just completely random. But that's what it is. What happens, for example, sodium hydroxide is added onto a beaker when there's HCl inverse indicator. What is the color? Well, if you're adding sodium hydroxide into a beaker, so if you're adding sodium hydroxide into a beaker, the beaker has a, let's just start the new, scroll down a little bit. Let's say this is HCl, yeah? So this is red. Why is it red? Because the pH is 1. And then when you start adding in sodium hydroxide, the color will go from red to orange, orange to yellow, you know, the whole yellow, the whole rainbow colors, red, orange, yellow, green, blue, purple, and then you just need to talk about, yeah, this is pH 1, pH 3, pH, yeah, this is yellow, pH 5, green's pH 7, this is 10, this is 13, for example, and this is H plus greater than OH minus. H plus is a little bit greater than OH minus. H plus is still greater than OH minus, but not that much. H plus is equal to OH minus, and then OH minus is bigger than H plus, and then OH minus is bigger than H plus. There you go, easy peasy. Excellent answer. What's the next one there? What's the percentage of people who fail science? Too high. Hunter, I've had you for 2 years in a row, buddy. I think it's fair for someone else to have a turn. Why does genetic have so much writing? You think that has much writing? Wait until you do some biology. Nucleotides, you will never be required to draw nucleotides. If they ask you to draw nucleotides, don't draw it. It's achieved. Don't bother. It's not something you're gonna lose. You know, it's worthwhile losing sleep for. Alicia, yeah, there are like 13 classes, 1 out of 13. There you go. There's 1 year where they had to label nucleotides. Yeah, that's just 1 year. Glad I'm dropping all sciences. Yeah, I mean, it's not for everyone. Not sure. Yeah. So for that enthalpy for the year 13, for that 1 year 13 that's in here, yes. So a spontaneous negative change in enthalpy, positive change in entropy is always spontaneous. Yes. Do I take 1 ACX? No, I used to, not anymore. Is science in year 11, year 12 hard? Yes. Yes, it is hard. I'll give you one example, yeah? You know the balancing equations in acids and bases? You know how that's excellence in level 1? And in level 2 chemistry, that's not even achieved. That's not even assessed. It doesn't even worth anything because it's so easy. So there you go. That's one example for you. And 99.99% of the course is new. Like you wouldn't have learned it before. We'll need to know sugar, phosphate, and base. Just it looks like this. It looks like this. Lump. This is sugar, base. This is, and then this is phosphate. Just remember that. That's not easy. That's not hard to remember. What's the easiest year 12 science? Well, that depends on you. If you what's the easiest science? Like if you're very good at math, physics will be for you. Like very easy. Like I always think of this like physics is a lot of mathematics, bio is a lot of writing, chemistry is in the middle. So you know it depends on what you're good at. What time should someone revise things or not? 2 weeks is enough. Yes. Do we need to know bases names? Well, all the hydroxide and all the oxide. There you go. William, get out of here. Kind of wondering how many trees in the QA. Yeah, well, they do, Sandwich Muncher. I'm pretty sure they use recycled paper. Yes, I'm pretty sure they do. 2019, question 2C. 2019. If you give me 1 second, 2019. Where the heck is it? Was it this one? Hello. Wait, that's not. That's a question 2C. Wait, is this it? Hello. Don't play this. All right, so there. If you want 29C, there. Done. Go look that. Go read that. Go watch that. 2019 question 2C. There. Can you do the base names, please? So hit just any hydroxide, like calcium hydroxide, sodium hydroxide, zinc oxide. So just anything with oxide and hydroxide. That's your, yeah. All right. Yeah, so if you're wanting like 2019, 2020, 2018 exams, is all on my YouTube channel. Just it's much more detailed in terms of explanation. If you just want to go on there and just watch those, they are a bit long, but I've been told that my videos are a really good cure for insomnia. Like say, puts people to sleep. Yeah. Yeah, I wish you guys all get E. If you get E, then that makes my time worthwhile. I mean, as long as you guys are happy with what you got, you don't necessarily all need to get E. Some of you will be happy with Achieve, some of you will be happy with Merit, some of you won't be happy unless you get an excellence. You know, everyone's at different places. Yeah. Is it okay if I do products minus reactants? Yes, you can, as long as you got given all enthalpy or formations. Is mechanics easier? well, do you like more writing or do you like less writing? Do you like math or do you like less math? So there you go. If you like more math, then you love mechanics. How easy is it to get achieved? It's too easy. It's a joke. If you get 8 out of 24, you passed. Oh, thanks, Santa. I'm a small content creator. Oh yeah. Looks like I'm gonna give you hiding tomorrow when I see you in class. Yeah, good luck, guys. What is the one thing that we should know? Well, sleep early, wake up, don't sleep, don't get up too early, have a nice sleep, make sure you're really well prepared, have some breakfast before you go in, and take a deep breath before you open the paper, and once you get started, if you get stuck with a question, make a note, move on, do another question, okay? Yeah, what does your GPA need to be to get into cellular science? Our thing is a little bit different. Every school is a little bit different. Hey, $1 donation. I think that's Tony from year 13. Money. Yep. So probably a good time for us to wrap this up. Maybe go to sleep. If there are any year 13s left here. Lily, which one do I think is the best for start with? Which one is your best standard? Which one are you best at? Start with that one. Alicia, they had a student standing on the bridge. We had a boy standing on it and it still didn't break. Yeah, $6 streams is a lot of work. If I were to do this for money, I would have quit a long time ago. One piece of advice for each paper. Well, again, depends on what you're looking for. Are you wanting to pass? Are you wanting to get merit? Are you wanting to get excellence? Hey, this guy is so rich. Tony, are you doing your 8 quiz exam tomorrow? You better be. Oh wait, you're probably not because you got achieved. Is over certificate endorsement more important than course endorsement? To be honest, Stacy, I don't think any of the endorsement matters too much. It's more just an acknowledgement of your achievement. It doesn't really mean much. Like say if you go to an employer and say, hey, I'm level 1 excellence endorsed, what does that mean to them? Probably nothing. Yeah, well, this guy has been oh my god, this guy's donating money to me. He sits right in front of me for Chem, and from all the crap that I've been taking from him this year, I think the money is well deserved. Can I do for each thing AME? Well, the best bit of advice is that for bio, learn your definitions. That will go across A to E. For Chem, learn the basics like atomic structure, electron arrangement, the pH colors, then you'll pass. And then if you can explain it, you can get merit and excellence. Mechanics, know what equation to use. That's been the most important one. Yeah. Last minute advice is that you need to go to sleep, and so you don't, oh my god, the 2 people asking for last minute advice for acids and bases and mechanics. So just acids and bases, just I don't know, just you gotta if you haven't grinded the exam papers, you should know where you're at. You should know where you're at. It's more of a if you're well prepared, you'll be ready for anything. Don't panic, okay? Just go into it. I assume it's going to be pretty fair. Like I'll give you one example that should put you a lot of at ease. I think it was last year's exam paper. We pretty much just copied the marking schedule from the year before because the questions are so bloody similar. Okay. Yeah. For the pH scale, how many greens are there? 7. Sahib, 7. pH 7 is green. Do we get into level 3 science in year 12? Do we take another year? Kingan, you only do one level 3 subject at level 2. So you can still do all your other level 2 sciences at level 2. Is genetics hard? Lots of writing, but no. Oh my god, Tony gave me another $10. Yeah, this guy. Does that mean you guys... No, see, this is... We did this 2 years... Was it 2 years ago or last year? They were... The questions were so identical, we just used the same marking schedule. It was literally the same questions. They just changed... I think they changed from calcium to sodium or something. We just changed like a couple letters, couple numbers, and then just use the same marking schedule. Yeah, pH 8 goes to blue. Yeah, it's just, yeah, it's unbelievable. Can we substitute E in for W? Like, wait, oh, work equal to energy. Work, Grace, is energy. Work is energy. Work, like how much work are you doing? So how much energy are you doing? Yeah. All examiners nice, or there's strict ones. Well, I mean, there's some horrible exam papers I've seen, like all the math exams. You know, people always cry about math. But yeah, Leon, go watch my other year 13 videos. Get out of here. Do you know definitions for mechanical? Not really. No. How do you keep the exam paper confidential if one of the writers? I haven't seen the exam paper. You think they'll let me have? You think they'll let me see the exam paper when I run the YouTube channel for tutorials? They're not going to show me the exam paper. Since you're going to mark them, I surely have to say it's easy. Look, I haven't seen the paper. I see it later than you do. You see them first, and I only get given them a couple of hours later. So I'm not an exam writer. Stop spreading lies. I'm a marker. Yes, I can tell you that because you don't know. You know, there's no conflict of interest here, but there are lots of teachers that mark. Yeah. Alrighty, I think everyone's pretty happy. 10:43, good time to wrap it up and for you guys to go to sleep. Yeah. Do you know if they're sending out exams back? Everything I'm pretty sure will be scanned. I'm pretty sure, science-wise, digital marking. Yeah. No, it's just being scanned. Everything's digital now. Yeah. Yeah, so it's all digital. Is DNA made of nucleotides? Yes, DNA is made of lots of little nucleotides. Can I give you a question? All right, hurry up, give me a question. Go on. You have 2 minutes. Well, no, I can't just give you E. Well, I'm not even telling what standard I'm marking, so there you go. You don't even know which I'm marking. Alright, see you guys. So off you go. Anyone else that's wanting to... Don't go to sleep at 5. You want to sleep early. Any last minute question before I wrap it up? Have a nice one, Alicia. No, we good. Can you explain the pH question? What is a pH question, Zainab? Yeah. As in power output of, calculate the mass. So, alright, here it is. No problem, Stacey. Bye. Alright, so if it's 115 or watts over 6 seconds, so what we can do, you haven't given me enough clues, James. Power equals work divided by time. So I know this, I can know work. Work is just going to be 1150 times 6 seconds. Is it 6 seconds? Wait, 6 seconds? Why did I write 5? So that's going to be work, and then I need to know distance. How high did the person climb? Use this number divided by the height, and then you go. All right. And so I'll do. Wait, is gene made of nucleotized DNA? A gene is a section of a DNA, and genes are made up of the nucleotides as well. So the nucleotides are the tiny blocks that build up the house. You can think of the gene as your living room, your kitchen, your bathroom, and then DNA is your whole house. There you go. The pH, the changes in the color. So there you go. This is the pH. So you go from red to orange to yellow to green to blue to purple, and these are the pHs and these are the ions. You know, just go through one of those exam papers on my YouTube channel that has it. Yeah. Kingan, did I mark your UGs? Yes, I did. You're welcome. All right. Sweet. All right, team. All the best for tomorrow. Don't sleep too late, and let me know how it goes in the Discord, in the NCEA Discord server, and all the best. Okay, all the best. All right. Have a good one, guys. I'm out. Good night.